Welcome, everybody. It's my pleasure today to introduce Dhananjay Bhaskar, and he'll be telling us about data-driven modeling and TDA of self-organized multicellular architectures. Take it away.
Thank you, Henry. Well, first of all, I'd like to thank the organizers for giving me the opportunity to present my work here today, and also for maintaining a really wonderful website and a YouTube channel with tremendous resources that have been extremely beneficial, especially to someone like me who doesn't really have formal training in algebraic topology. Before I kind of get started with my talk, I'd like to kind of give you the two key take-home messages that I want to deliver today. Well, first message is that simple mathematical models are great fun to play with, and they can generate immensely complicated behavior. And the second message that I want to deliver is that topological data analysis in combination with a little bit of machine learning has a tremendous potential in analyzing the complicated behavior that comes out of these simple mathematical models and also from experimental studies in cell and tissue biology. So I have organized my talk in terms of difficulty. So we're going to start with a very simple problem where we are going to apply TDA to a homogeneous population of cells. Then we're going to increase the level of difficulty by allowing ourselves multiple cell types in our population. And then finally, we are going to incorporate temporal dynamics into our data. And so to begin with, I'm going to take some inspiration from cancer biology and talk about epithelial to mesenchymal transition and how this is important for cancer biology, but also has some implications for developmental biology. So the epithelial to mesenchymal transition is basically a phase transition that is observed in multiple biological phenomena, including embryo development, wound healing, and cancer metastasis. And this phenomena basically refers to a phenotypic transition where highly compact, well-organized cells lose their cell-to-cell adhesion, they lose their compact cell shape, and they become highly motile, acquire a mesenchymal phenotype, a proliferative phenotype, and they start invading into their surrounding environment. And this process is associated with the expression of various transcription factors, cell-to-cell signaling, which basically allows these cells to start cleaving the extracellular matrix that surrounds them and invade. So you can see how going from the epithelial phenotype to the mesenchymal phenotype is important in the context of cancer biology. And quite interestingly, once these cells are able to invade into their surrounding environment and enter into blood vessels, they eventually manage to find secondary sites to colonize. And there they undergo the reverse transition from the mesenchymal state back into the epithelial state. And so this process is very, very important, not just for tumor initiation and metastasis, but also for colonization of secondary tumor sites. Here's another depiction of the same process. This is one of the hand-drawn schematics from Peter Friedel. Again, you can see that the epithelial phenotype has this nice compact morphology where cells are quite well organized. And once they go through this transition, these cells lose their shape. They start cleaving through this extracellular matrix by expressing these matrix metalloproteases, and they start invading. And so in the lab, we can study this using confocal microscopy. So I just want to quickly show you what this looks like when you actually look at this process under a microscope. So on the left here, we have epithelial type human mammary cells. These are cells from the human breast. And you'll see here that they are forming this nice compacted clusters. Well, if we start expressing snail, which is a transcription factor that allows these cells to undergo this EMT transition, then you'll see that these cells are no longer forming nice compacted clusters. They are moving around. They're highly motile. They're proliferating a lot. And they're going to continue to proliferate until they form a confluent monolayer. What's quite interesting is that this process can be stalled if we reduce the amount of epidermal growth factor in this medium. So if we get rid of EGF, what you'll find is that these cells are going to acquire a mixed phenotype where they are somewhere in between the epithelial and mesenchymal state. You'll see that they are forming some kind of cluster. In fact, they're forming some kind of spanning network, which is somewhere in between this compacted cluster morphology on the left and the highly confluent morphology and the highly motile phenotype on the right. And so this, we think, is a great thing for stopping the metastasis of cells from the primary tumor site, is if we can starve that tissue of EGF, then we hope that these cells are going to not be able to invade into their extracellular matrix. So that's kind of the biological context in which we want to study this process. And we can, in fact, segment these images and start to develop a mathematical model. And so I'm not going to go into details for how the segmentation works. Suffice it to say that if you have a label on the cell nuclei, as well as a cytoplasmic label, you can then segment both of these channels. And then using a little bit of image processing, you can segment out not just the shape of individual cells, but also the shape of these clusters. And then you can quantify this cluster morphology using something as simple as fractal dimension. And so this procedure of cell tracking and cell segmentation gives us a lot of information that we can then use to develop a mathematical model. And I particularly like simple models. So this is the model that I developed. It basically consists of like 3 different equations. So we are going to represent cells as rigid bodies, these spheres. And the main governing dynamics are controlled by 2 parameters. Firstly, we have this polarization force P, which is a random motility force that we apply to these cells. And secondly, we have a force F, which basically governs cell-cell interaction. And this force F depends on a kernel U, which looks a little bit like this. And so this kernel U basically governs how attraction and repulsion works. And we have short-range repulsion to prevent these cells from getting too close to each other, and slightly longer range attraction that allows these cells to aggregate and form clusters. So the strength of attraction and repulsion is governed by parameters CA and CR, and the length scales for attraction and repulsion are governed by parameters LA and LR. So it's quite a simple model of this highly damped mechanics with just 4 parameters, CA, CR, LA, and LR, that we can calibrate using our image segmentation and cell tracking algorithm. Okay. So once we have a calibrated model, let me show you what kind of dynamics it can generate. So here I'm going to show you 3 movies at the same time. On the left, we are going to have low adhesion force between these cells and high polarization force. On the right, we are going to up the adhesion force and decrease the polarization force. And what I hope you can see quite clearly in these simulations is that on the left, the cells are highly motile. They do not form stable clusters, and they end up forming a confluent monolayer. On the right, they form these compacted cluster morphology. And in the middle, we have this branching spanning network. So hopefully, qualitatively, at least, you can see the similarity between our simulation results and the experimental results I showed you before. I'd also like to mention that we have periodic boundary conditions here, as well as the color scheme basically depicts how many neighbors a cell has. So if a cell is isolated, it's shown in red. If the cell has fewer than 4 neighbors, it is shown in blue. And if a cell has 4 or more neighbors, then I'm going to plot that cell in green. We also implemented cell proliferation. So cells that are in red and in blue, that are cells that have fewer neighbors around them, are able to proliferate. And the time to proliferation is again calibrated using the cell tracking and cell segmentation algorithm from the experiments. Okay, so qualitatively, at least, we see some similarity. Quantitatively, if you measure the number of clusters over time, cluster size over time, or if you look at the cluster shape by plotting the radius of gyration versus cluster size, which basically gives us the fractal dimension, I hope you will agree that our simulation results match very nicely with the experiments that I showed you previously.
Okay, so quick question. Sohail in the chat was asking if you could describe again how the parameters, I think on slide 7, were related to the animations on slide 8.
Sure. So the parameters that I'm going to vary is the magnitude of the polarization force P. So if you recall, this polarization force is a random force that is applied to a cell. So that's basically what controls this kind of Brownian motion in the absence of other cells. And secondly, I'm going to vary the adhesion force. So adhesion is governed by the parameter Ca. This is the strength of adhesion, and that's what I'm kind of varying. In fact, I'm varying Ca over Cr. I can look at the ratio of these two parameters, and I can tune how adhesive these cells get. One thing I did not really mention here is we don't have, this force is not applied up to distance infinity. We do in fact have an indicator variable here that basically says that this attraction force is only going to come into effect if the two cells are within 1.5 distance of each other. And if they get too close, then of course they're going to repel. So that is basically what gives us this cluster formation. Mm-hmm. Okay. Okay, so now that we have a well-calibrated model, we can run this on a cluster and start to perform parameter sweeps. So here again, I'm going to vary these two parameters, the propulsion force or the polarization force on the y-axis and the adhesion strength on the x-axis. And the heat map here basically shows you the density of the population. And so as you would imagine, at low polarization and high adhesion, you have the formation of these clusters. At high polarization and low adhesion, the cells are going to stay individually. They're going to be individually dispersed, and any kind of clusters that they form are not going to be stable over time. They're going to break apart. So this is the set of simulations where I don't have proliferation turned on. So the population size stays constant. And we can also, if we have proliferation turned on, we can see a similar kind of phase diagram where you can go from a clustered state to this branching state at higher proliferation and lower adhesion values. One thing that you would note here is that this distinction, like this phase transition, is not really very clear if we plot this order parameter of local density. In fact, it's quite hard to come up with order parameters that are going to tell us exactly where this phase transition is going to take place. And that is basically what motivated us to use TDA to see if we could do a little bit better than order parameters at identifying these phase transitions. And so I'm not going to go into TDA. I'm going to assume that the audience is very familiar with TDA, but I'll just give you the punchline here. So on the left, we have individually dispersed cells. In the middle, we have the spanning network. And on the right, we have the compact cluster morphology. We don't want our TDA-based algorithm to be sensitive to the population size, right? Because we have simulations with proliferation turned off and proliferation turned on. And ideally, we want a methodology that works even if the cell number changes across simulations. And so therefore, we decided to use just dimension one homology to quantify differences between these simulations. And the way we do this is we compute persistence diagrams, of course, for dimension one homology. And we find the following observation. We find that in the compact clustered morphology, there are very few loops, but the loops that are present are going to be quite big. And these are the loops that are going to basically encode the empty space between these big clusters. If the cells are individually dispersed, on the other hand, we find that there are lots and lots of loops present in our data. So basically, there's quite a lot of empty space between these cells, but that empty space is not going to be as big as the empty space that we find between our clusters on the right. So intuitively, you can imagine that the presence of topological loops tells us how much empty space is present between these cells in these different simulations, and therefore, we have a good chance of being able to classify them into distinct phases if we use dimension one homology. And this dimension one homology computation is not going to be sensitive to how many cells you have in the simulation as opposed to dimension zero homology. So we can come up with a methodology that's agnostic to population size. And that's exactly what happens, right? So here we have we have all our simulations on the X and Y axis. So this is a giant kind of distance matrix. And so what we did here is we computed Wasserstein distance between the persistence diagrams of all of these simulations, only looking at dimension one homology. Of course, this is going to be a symmetric matrix. And in fact, we can compute each entry in this matrix in parallel. And you could do this on your computer. If you have lots of simulations, you can actually do this on a cluster as well, which is what we did. So once you have this pairwise Wasserstein distance matrix, then you can use some unsupervised machine learning to find clusters, and then take those clusters and map them back onto the phase diagram. So this is the phase diagram I showed you previously, where we had cell proliferation turned off. And if we actually find clusters using our pairwise Wasserstein distances and map them back onto the phase diagram, we find that there are 4 distinct phenotypes. There are clustered simulations. There are simulations that show some branching with some small clusters present. We have branching simulations, and then simulations with individually dispersed cells. And this ordering of going from the clustered state to the individual state intuitively makes sense, because this is what you would expect as you reduce adhesion and increase the random polarization force. So far so good. You can apply the same technique now for simulations where proliferation is enabled. And you actually end up with a nice phase diagram again, where you go from the clustered state to branching with clusters to the branching phase. So using this methodology, we were able to identify the phase transitions in a manner that is a little bit more satisfying than simply looking at some kind of order parameter. It's quite clear where this phase transition happens. The entire methodology is obviously unsupervised. So you don't have to sit down and think about what kind of order parameter you would use. You can simply apply topology, dimension 1 topology, and let a little bit of machine learning tell you where the phase transition happens.
Dhananjay, let me interrupt with a question, if that's okay.
Sure.
So Brett Jefferson types, could it be that the proliferation effect can be thought of as a robustness check? He says, sort of sounds like yes. I mean, is proliferation related to robustness as you add more points?
Absolutely, yes. So if we are, as I'm going to go into in this slide, especially if you're working with experimental data and not just simulation data, you not only have cell proliferation, but you might lose track of a couple of cells. They might go in and out of your field of view, or simply the nuclear label might disappear sometimes, especially when the cell is going to divide. And so you want this robustness to population size built in. And that's precisely what we get if we just look at dimension 1 homology. Okay, so now that we have a way of identifying these phase transitions using topology, what we want to do here is we want to map experimental results onto a phase diagram that's built using simulations. The reason
Is it okay if I ask another quick question?
Sure.
Just a clarifying question. So on the plot of the local densities, I understand that maybe that's an average over time. But if are you taking an average of persistence diagrams over time? Because in the plots that you showed, the configuration is evolving in time.
Yes, that's a wonderful question. So no, right now we are keeping things very simple. So I promise you that I'll start with a very simple problem, right? So I'm only looking at the final configuration of cells. So this is the configuration I find at the end of the simulation, at the final time step. And it's actually quite remarkable, considering that I'm not incorporating any temporal information, how well this classification actually performs on a single static snapshot. And as we progress through the presentation, I'll talk a little bit more about when incorporating temporal information might be more useful.
Thank you.
So in this slide, what we did is we took our experimental results and we computed the persistence diagram from cell nuclei positions in the experiment. And then we computed Wasserstein distance between this experimental configuration against all simulations in our model. And so wherever that Wasserstein distance is minimized, that's basically the match that we find between the experiment and the simulation. And so just as an example here, if you perform an experiment in growth media where there's lots of EGF present, well, with EGF, these cells are going to become polarized, and therefore you expect this simulation well 24 and well 1 to land at a higher proliferation. So as expected, these two simulations show up somewhere over here. where these things are forming branch morphology and they have higher polarization. If you then decide to inhibit EGFR signaling with a drug like gefitinib, you expect that they are not going to have such a high level of polarization, and that is exactly what we find is with well-3, our experiment lands somewhere over here. So you can now imagine tuning your experimental parameters by mapping these things onto the phase diagram and trying to figure out that as you change the concentration of gefitinib, how that simulation is going to map across this phase diagram. So basically, this is a powerful technique for taking parameters that we can control in the lab and mapping them onto a very simple phase diagram constructed from simulations. It allows us to map experimental parameters onto simulation-based parameters, and it allows us to figure out what sort of experiment we should perform next if we want to see some kind of phase transition. It also allows us to quantify how well these different drugs for EGFR signaling, to inhibit EGFR signaling, are working. One of the previous questions that came up was the robustness of this approach, and that is basically what I get into with this slide. So here we take our individual branching and clustered phenotypes, and we simply start deleting a bunch of points on the left, and on the right, we start scaling. So we are zooming in more and more into the simulation, and at the bottom, I plot the Wasserstein distance between these simulations. And so what you can appreciate here is that just by eye, a human can tell these things apart up to around 150 removals. And in scaling, you can tell them apart at around 2.5x scaling. And that's precisely what the algorithm is capable of doing as well. So the ability of the algorithm matches our human ability to distinguish these things with respect to these kinds of perturbations. Now, I do realize that there's going to be a lot of mathematicians and physicists in the audience, and I've done the cardinal sin of plotting these phase diagrams without specifying units. So I just wanted to mention here very briefly that you can, in fact, take this model and non-dimensionalize it. So you can come up with a Peclet number and adhesion polarization ratio, and then you can replot the diagrams with these non-dimensional parameters. It's just not convenient to look at these kinds of diagrams. It's much better to look at the phase diagrams I was showing you before. But just to cover all bases, I wanted to show you can non-dimensionalize the model. So to summarize this part of the talk, we come up with a very simple mathematical model that consists of two key ingredients: random polarization and cell-cell adhesion, which is able to capture the dynamics of cell aggregation. We were able to identify phase transitions in simulations with and without proliferation by computing pairwise Wasserstein distances of dimension 1 homology. And this is done in a model-agnostic manner. We found that our overall approach was robust to random particle removal and spatial scaling. And we were able to beat the performance of order parameters in this phase transition identification task. Okay. Some of the limitations of the model is that we were working with a homogeneous population consisting of a single cell type, and our pairwise Wasserstein distances were computed at the final time step. So in other words, we did not consider temporal dynamics. And so in the next part of the talk, I'm going to address these limitations. And so switching gears now, we are going to go from cancer biology more towards developmental biology, where I'm going to talk a little bit about pattern formation in embryonic cell lines, where we have multiple cell types. And I'm going to show you how this methodology generalizes once we have different kinds of cell-cell interactions with different cell types in the population. So just a brief primer. If you take two different embryonic cell types, let's say you're working with a chick embryo, you take some retinal cells and some kidney cells or some other type of cells, and you mix them up in a blender, then what you're gonna get over time is that these cells are going to automatically configure themselves based off of their adhesion properties. So in certain cases, you will find that the two cell types are going to separate from each other and form separate clusters. In other cases, one cell type is going to get entirely engulfed within another cell type. In a different configuration, they are going to get mixed up and form some kind of checkerboarding pattern, or sometimes they might just stay individually dispersed. So this has been well known for a long time, but how we transition from one state to another has not really been well understood or comprehensively kind of studied until now. And so this is just an experimental picture where this experiment was performed in 1971, where Armstrong took these chick embryo cells, he mixed them up, and then over time, you will find that one cell type got entirely engulfed within another cell type. If you want to do this experiment at home and you live close to a beach, you can go and find 2 different species of living sponge in the ocean, bring them back home, put them in a blender, and let it sit. And you will find that they will automatically separate themselves out and create 2 different organisms. That's kind of a cool experiment to perform at home. And so we can model this. It actually takes just a small change to our model. So we're still going to have our random propulsion force. And now our cell-cell interaction force is going to depend on the cell type. So the way we are going to encode this is we are going to introduce this parameter gamma, which controls the strength of cell-cell adhesion. And this parameter gamma is going to now depend on the cell type of cell I and cell type of cell J. So basically, we are encoding differential adhesion, where I'm going to show you simulations with blue cells and orange cells, and I can independently tweak how strongly blue cells adhere to each other and how strongly orange cells adhere to each other, and what the adhesion between blue and orange cells is going to look like. And it's quite easy for me to just kind of show you the result. So for example, if you perform a simulation where blue-blue adhesion is high, but blue-orange adhesion and orange-orange adhesion is low, then as expected, blue cells form clusters and orange cells stay individually dispersed. If you have high blue-blue adhesion and high orange-orange adhesion, but no blue-orange adhesion, then they are going to form clusters that are going to stay separated. If you instead increase the blue-orange adhesion, then as you might well imagine, they're going to try and maximize the interaction between blue and orange cells. And that's going to result in the formation of these hexagonal patterns. We know that hexagonal is the best energy configuration for maximizing the adhesion between 2 cells. And in all other intermediary kind of parameter values, you get lots and lots of cool pattern formation, including things like stripes and spots, which might remind you of patterns on the skin of some fish. So I'm going to talk a little bit about these zebrafish skin pigmentation models a little bit further down the line. So this is just a foreshadowing what's to come next.
And a quick question from the chat. Do these different arrangements have different implications for life or quality of life?
Yes. So if you take a embryo and you experimentally perturb the adhesion properties of a single cell type, that is going to have major implications in terms of these cells being able to organize themselves and form organs. And so that embryo could become entirely non-viable, or it could become an embryo that has severe deformities going forward. So so any kind of problems in cell differentiation and the adhesion properties of these cells early in embryonic development can have serious developmental consequences for the embryo. Okay, so now that we have 2 cell types, or more than 2 cell types in general, our previous strategy of simply computing Wasserstein distances between the simulations is not going to work. It's not obvious how we should combine these Wasserstein distances. So for example, if I want to compare these 2 simulations, I can compute the Wasserstein distance between the persistence diagrams computed from just blue cell positions, just orange cell positions, and blue and orange positions combined. So I get 3 different pairwise Wasserstein distance matrices, but it's not obvious how to combine them. And computing the multiple Wasserstein matrices across a large range of simulations is going to be computationally expensive. So we instead rely on persistence images. And so we compute both dimension 0 and dimension 1 persistence images by taking our persistence diagram and dropping a Gaussian at each point in the persistence diagram. And basically summing them up to compute a dimension 0 image, which I'm going to plot as a 1-dimensional plot, and dimension 2 persistence image, which I'm going to plot as a contour plot going forwards. And we compute these images for just the blue cells by themselves, the orange cells by themselves, and the blue and orange cells combined together. So we are in fact computing 3 different persistence images for these simulations. And then we are going to feed that into a machine learning model to be able to see if we can find these phase transitions. So just as an example, I show you here 2 simulations, 2 simulation results, one where blue cells are forming clusters and another one where we see some kind of hexagonal patterning. And so the dimension 0 image looks a little bit like this, where, as you can imagine, we have one peak for merging events between these cells, and then 3 different peaks corresponding to merging across these clusters that happens at a higher epsilon value. In dimension 1 homology, we see that there's some small loops within each cluster, and then one larger loop here that exists across these clusters. We also see some loops in orange, which are going to be bigger than the loops in blue because there's more empty space present between our orange cells in the simulation. So just to be clear, the middle plot is the dimension 0 persistence image, and the plot on the right is the dimension 1 persistence image. Similarly, for the hexagonal configuration in dimension 0, we find that the blue cells are quite close to each other, so the peak appears at a lower epsilon value, and the orange cells are further apart from each other, so that peak shows up more towards the right. And we find that the loops in orange are bigger, so they start at a higher epsilon value, but the loops in blue are more persistent, which is quite obvious once you look at the configuration. These blue cells are in fact forming loops that are going to exist for a large range of epsilon values.
Ask a quick question.
Sure.
So the one-dimensional persistence images I'm able to parse. So you're looking at the the contour maps of these sum of Gaussians. Somehow I'm having trouble getting my mind around the zero-dimensional.
The zero-dimensional persistence.
So are all the birth times equal to 0 here? Or can you explain this one a little bit?
Yes. Yeah. So because in dimension 0 homology, all our points in the persistence diagrams are always going to be at epsilon equal to 0 on the x-axis. Yeah. So it doesn't make sense for me to convert this into like a 2D image. Right. So once I drop Gaussians on these points, I can simply take the intensity profile along this axis.
And then you're just rotating it.
And then I'm just rotating this. Yeah. Exactly. We are weighting the Gaussians by distance from the diagonal as well, because we figured that points that are further away from the diagonal are going to correspond to more relevant features for this problem. So I hope that kind of clears up why the dimension 0 image looks like this. And as I mentioned before, we're going to simply take these images, we're going to concatenate them together, and then feed them into a model to perform unsupervised classification. And so this is just to show that we had 512 simulations where we varied the orange-orange adhesion. So we had 8 different values for orange-orange adhesion, 8 different values for blue-blue adhesion, and 8 different values for blue-orange adhesion. So we are going to come up with a 3-dimensional phase diagram by varying these 3 different adhesion parameters. And you can actually take the model and figure out where exactly you expect to see collective behavior versus individual behavior with a simple calculation of the attraction force divided by the magnitude of the random polarization force. This is not really super important. This is more important. So this is basically the result that we got. You take all those persistence images, feed them into a model, run clustering, and you end up with this cube. And in the cube, we can identify all the different phenotypes. And so this includes individual clusters for blue cells and orange cells, individually dispersed cells, blue clusters, which contains very tiny, small orange clusters embedded within, or like orange clusters that are completely surrounded by blue cells, or spotted patterns, or stripy patterns. And so this phase diagram basically tells us how these different distinct phenotypes are all related to each other in terms of cell-cell adhesion. And to the best of my knowledge, this is the first time that somebody has managed to actually figure out how these configurations are related in a kind of comprehensive study. And so we can take this cube and we can look at different slices along different axes. So as you would expect, for example, here, if our blue-orange adhesion is 0, then if your orange-orange adhesion is high, orange cells form clusters and blue cells stay dispersed. Blue-blue adhesion is high, blue cells form clusters and orange cells stay dispersed. If both blue-blue and orange-orange adhesion are high, then both of them form separate clusters that do not mix. And if both of them are low, then all cells stay individually dispersed, and so on and so forth. So the result we get from our clustering makes sense to us intuitively by examining these adhesion values. And we did, in fact, not just work with persistence images, but we also computed persistence curves and order parameters. So what I'm showing you here are dimension-reduced topological features. So on the left, we have dimensionality reduction performed using UMAP. In the middle, we use a manifold learning technique called FATE to plot these in 2 dimensions. And on the right, we use PCA. And what's quite obvious here is that order parameters are not able to distinguish between these different configurations. Persistence curves perform a little bit better than order parameters, and persistence images perform very well. And in fact, if you look at these persistence images, you will see that, for example, these white labeled cells are adjacent to the pink labels and the blue labels, as well as the yellow labels. So you expect these simulations that I've labeled in white to be adjacent to the simulations in pink, blue, or yellow in the phase diagram. And that is exactly what we find. So you can, in fact, recover relationships between these different simulations, even with this just low dimensional embedding produced using this unsupervised manifold learning algorithm. And we can take these results. So I have all those FATE plots here on the left, and we can see how these simulations vary in terms of the actual adhesion values. And I hope you'll agree here that with persistence images, we are, even in 2 dimensions, able to separate out simulations with low blue-blue adhesion or low orange-orange adhesion or low blue-orange adhesion. That's basically to say that if we just plot these simulations in 2 dimensions, we can find that there are sections of this plot where we have simulations where blue cells are forming clusters, another section where orange cells are forming clusters, another section where blue-orange cells are going to mix together and maximize their interactions with each other. So that's the agent-based model that I developed. And next, I want to quickly talk a little bit about zebrafish. So if there's any questions regarding the work I just presented, perhaps this would be a good time to pause.
I have a quick question, actually. Are the configurations the same regardless of the initial conditions? Like you take a bunch of points, does the final configuration not depend on which points you start with?
Yes and no. So you can kind of construct an adversarial attack in the sense that you can start with the initial condition where, let's say, all blue cells are on one side and all orange cells are on completely the other side, but the adhesion parameters are set up so that they should form a single cluster that's well mixed. And they will form that single cluster that's well mixed. You're just gonna have to wait a long time as the simulation proceeds. So we run these simulations up to 5 million time steps because we are extremely paranoid about whether or not we have reached steady state. And reaching steady state is easier in some parameters and not so easy in other parameters. And if you end up like getting stuck in a local minima, if we think of this as like an energy minimization problem, then it can be sometimes quite hard to get out of that local minima and reach that global, most stable configuration. So you can certainly make life difficult for yourself if you start with the initial condition that's set up to kind of make your simulation job much harder in the sense of getting to that globally optimum kind of configuration. But they will that configuration.
But there is some kind of theoretical principle that says that there generically some kind of limit configuration.
Yes, so that is work work that started by Steinberg. So he was the one who originally proposed this differential adhesion hypothesis to relate pattern formation to these adhesion parameters. And that hypothesis was then modified by work by Broadland, and he come up with the differential interfacial tension hypothesis, where he provides inequalities, which if you satisfy those inequalities in terms of your initial conditions, then you are guaranteed to end up at that configuration. There has been some work in that direction, but I don't know if that applies to all configurations. There are certain configurations where you can actually prove that it will reach that final state.
Thank you.
That's my understanding of it.
Very quick question. Can I ask?
Well, let me call on Brett
sorry.
first, if that's okay, Hussein, just because he had his hand up. But I'll call on you next. And Dhananjay, this is part of your question period, not your presentation period. So don't. But Brett, why don't you go ahead with your question, and then we'll take Hussein next.
Great, thank you. Just to make sure I understood the kind of batch of results, for that mixing result, you computed the persistence image for all points regardless of blue-orange kind of coloring, and you didn't worry about, let's say, computing the persistence for blue only and then the persistence for orange only and then combine, right? So it's the former rather than the latter, right?
Okay, so are you referring to these results here in terms of these classification results?
Yes.
Okay, so I computed persistence diagrams separately for just the blue cell positions, just the orange cell positions, and both in blue-orange cells regardless of cell type. So I computed 3 different persistence diagrams for each simulation. And again, I'm only looking at the final time step, so the very final configuration in the simulation. I compute 3 persistence diagrams, and then I convert them into persistence images, which I concatenate together and then feed them into the FATE algorithm or UMAP or some other dimensionality reduction technique, and then perform unsupervised classification on that embedding to come up with this classification result.
Okay, so this is the concatenation of like dimension 1, and then concatenate with dimension 2, and then concatenate with dimension 1 and 2 considered as just points regardless of that dimension 1, dimension 2 categorization.
Oh yes, that's another important thing I should mention. So I am concatenating persistence images corresponding to just blue cells, just orange cells, and blue and orange combined. But these 3 persistence images that I'm concatenating are only dimension 1 homology, because again, we want to not be sensitive to population size here. So I'm only looking at dimension 1 homology. In the zebrafish work that I'm going to present next, I will look at both dimension 0 and dimension 1 homology. But this result was computed using only dimension 1 homology.
Great, thank you.
We'll take one more question now. So Hussein, please.
Thank you very much. Very quick question. A question about the modeling. Usually in this phase transition models, the temperature is important, but I think for biology it is not important. Sorry to ask. The temperature.
Yes. So if we were doing like a model of like Janus particles or something like that, then I agree. Temperature is an important parameter. For the purpose of this, for the purposes of coming up with a data set where we could kind of validate this methodology, we did not consider those kinds of particle type models. And so in developmental biology, it's common to have the sort of model that I have. You can of course make it a little bit more sophisticated, so you can have cells that are deformable and so forth, but we wanted to perform lots and lots of simulations, and therefore we elected to use a very simple model. We have some work where we have applied this technique to the sort of models that you're mentioning, and it performs well in those conditions as well.
Thank you very much.
Okay, wonderful questions. Okay, I'm gonna now proceed and talk a little bit about zebrafish skin pigmentation. And so you can find designer zebrafish on the market today. You can find a zebrafish that suits your liking. You can get like different spotted patterns, stripy patterns. And in fact, you can take a zebrafish, and if you have access to a laser, you can ablate some of these cells. And actually, we know the underlying Turing mechanism that leads to this kind of pattern formation. So you can, in fact, take a zebrafish that has a stripy pattern, and with a small amount of laser aberration, you can convert it into a zebrafish that has a spotted pattern at home. Not something I would recommend that you do, but it is possible. This is an interesting system to study. There's lots and lots of models that already exist. The one that we worked with is called the Bullara-Dedecker model. And this is an on-lattice model where you have all these lattice sites, and you have certain rate of birth for these xanthophores, which are the yellow colored cells, and a birth rate for melanophores, which are the black colored cells. You have certain death rates for these things. And then you have interactions between a melanophore and a xanthophore. And you can take this on-lattice model and develop a continuum model and actually prove that this continuum model is capable of the Turing bifurcation, which basically tells you that is going to result in this kind of pattern formation. And so the objective here was to run these simulations and then compute topology of the patterns that result from the simulation, and see if we can solve an inverse problem where we take the simulation result and we try to identify the parameters that went into this model. And so for the purpose of this task, we kept all parameters fixed except this parameter H, and this parameter LX. And so if you just vary H and LX, these are the different kind of phenotypes you get. And if you actually apply the methodology that I just described, you can easily classify these simulations into what I'm going to call stripe simulations, sinusoidal stripe simulations, spots, and checkerboard or a labyrinth pattern. So in terms of just figuring out which phenotype is which, that is easily doable with TDA. But the question is, can we go a step further? Can we infer geometry? Can we infer the geometry of these stripes and spots using TDA? And then can we go a step further and actually infer the model parameters using TDA applied to these simulation results? And so to kind of look into this, what we did is we developed a simple generative model of stripes where we control the width of each stripe. So we are basically sampling these melanophore positions using a normal distribution with some sigma. We control the separation between stripes. We also control the amplitude and the frequency of the sinusoidal patterns. Okay, so we have a very simple kind of generative model to create these patterns, and we compute its persistence image. And this time I'm talking about dimension 0 persistence image. And we can then quantify what that persistence image looks like by measuring the intensity of these peaks, as well as the width and the location of these peaks. And so for the stripe geometry, you will always find two peaks. The first peak corresponds to merging between the melanophores within a stripe, and the second peak corresponds to merging of these melanophores across stripes. And so if you then run this kind of experiment and vary parameter, for example, here I'm varying my stripe width, you can then figure out how the peak location, the width of the peak, and the intensity of the peak changes as you vary that parameter. And then you can build a simple kind of regression model that takes that persistence image and predicts what the stripe width of this zebrafish is going to be. So this is an experiment that shows us that the peak positioning basically is similar to the interparticle distance for peak number 1 and the stripe separation for peak number 2, as you would expect. And that the width of the peak also changes as you would expect. So for example, in this case, as the stripe width increases, we expect the first peak to get wider because we now have to merge across more cells that are further away from each other. So it's going to take many different epsilon values to perform all the merges in dimension 0 homology across this, whereas the second peak is going to get thinner because these cells across stripes are getting closer and closer to each other. And so you can repeat this experiment also by varying the amplitude of the stripe, or you can delete cells within a stripe, or you can start scaling these simulations. So you can vary the number of stripes and also the distance between these cells by zooming in into the center of the simulation. And in all cases, the positioning of the peaks, the intensity of the peaks, and the width of the peaks changes as you would expect, which basically tells us why we are able to take these persistence images and compute stripe geometry or spot geometry using topology. And so I didn't really go into the details of that work, but I just want to kind of give you the summary in that using TDA and the methodology that I described, we can identify the distinct phenotypes, so stripes, spots, sinusoidal stripes, and labyrinth patterns quite easily. We can predict the geometrical parameters of both stripes and spots using TDA, but the recovery of model parameters cannot be performed to a high degree of accuracy using TDA. And that basically has to do with the fact that as you vary these model parameters, H and Lx, the time it takes to form a certain configuration is important. So if you want to disambiguate between these parameters, we have to incorporate temporal information before we are able to do that. And so just to summarize this part of the talk, we use TDA to classify pattern formation in heterogeneous populations. We used Euclidean distances between persistence images to perform this computation. We found that persistence images outperformed smooth persistence curves. I didn't show you these results, but they also outperform Euler characteristic and persistence landscapes on this task. And we found that we were able to recover different phenotypes as well as geometry, stripe and spot geometry for zebrafish simulations, but recovery of model parameters requires temporal information. And so I hope I have convinced you that we have overcome one of the limitations that I described previously, but we still haven't considered temporal dynamics. And I'm cognizant of the fact that I only have 5 minutes left. So I unfortunately don't have the time to tell you how incorporation of temporal dynamics can improve classification accuracy in our barcode. I will simply point you to a paper that I worked on with Lori Zeigelmeyer and Chad Topaz and a few other students in MRC, where we simulated a Dorsonia model of animal swarming. And we precisely tried to solve this inverse problem of looking at the collective behavior and trying to identify the parameters that went into that model. And for this, we incorporated temporal information using Crocker plots to do this. I'll just point you to this paper. And so to end, I'm quickly going to describe some ongoing work where we are incorporating temporal information to study lumen formation in 3D cancer spheroids. This is a very interesting problem. So if you actually build one of these cancer spheroids in the lab, especially with the breast cells, you will find that in 3D microscopy, these cells are invading outside, but also they are forming these lumens that are within this spheroid. And this lumen formation gets disrupted the more mesenchymal these cells get. So if you are looking at a normal breast tissue, they're going to form perfectly nice lumens in 3D. But if you then trigger EMT, then these cells are going to start moving away from those lumens, and that lumen formation is going to get disrupted. And that's one of the key kind of hallmarks of tumorigenesis in human mammary cells. And so we basically track these cells over time, and we computed a topological barcode at each time point. And then from the dimension 2 topological barcode, in this case, we computed cycle representatives to identify nuclei that are immediately adjacent to a lumen. And with a little bit of help with some heuristics, because these generators are not geometrically optimal, after incorporating a few heuristics to make sure that we find cells that are immediately adjacent to the lumen, it is possible to track which cell belongs to which lumen. So this is basically a preliminary result where we have labeled the cells in these spheroids as unassigned if they do not belong to a lumen, or we have labeled them in green or red or blue, depending on which one of these three lumens they belong to over time. And with these generators, you can actually track how cells move from one lumen to another. And in this particular case, we found that there's one lumen in the middle of the spheroid that exists throughout time. There's a lumen at the top, which exists at the beginning, but then gets destroyed as these cells start invading outside. And there's a new lumen that forms in this area towards the end of the acquisition. So we hope to incorporate temporal information and to also compute the cycle representatives to study this lumen disruption, especially in the context of breast tumors. And with just 2 minutes left, I will just show you one more problem that's of interest to us going forwards, is that if you look at these spheroids earlier in time, you'll find that they undergo this complicated rotational motion. So this is a spheroid, and you see that these cells are kind of going some kind of rotational motion, and this problem has not been very well studied. What we know in terms of the biology is that this rotational motion is important for the assembly of a basement membrane around these cells. And so once this basement membrane gets established, the rotational motion stops. But if you then experimentally destroy that basement membrane, the rotational motion restarts. And if you actually stare at these microscopy images for a long time, you can identify that there are leader cells and follower cells within this kind of rotational motion. And it's of interest to us to figure out how those leader cells are different from the follower cells. And so some ongoing work that I'm involved in is to develop a very simple mathematical model, which I'm not going to describe, but basically you have collective motion on a sphere, and then we are using topology to figure out who the leaders and followers are in this model over time, because the leaders are going to change, the cells are going to divide. So this is actually quite a complicated problem to figure out. If you have some ideas for doing this, I'd be happy to hear them after the talk. And with that, I'm just going to conclude with a few future directions that I'm super excited about. As I mentioned, identification of leaders and followers in spherical collective motion, studying collective motion on these tissue scaffolds that are just experimentally being developed now, and also incorporation of not just temporal information, but also computing multidimensional persistence modules, especially in cases where we have cell proliferation. We might want to consider like a density distance filtration because that's going to be a little bit more informative than what we have currently. And finally, in terms of recovery of model parameters, I was recovering the adhesion parameter and the polarization force parameter, but one can imagine using more sophisticated physics-informed machine learning together with some topological priors to learn that kernel U that governs cell-cell interaction in these simulations. And so with that, I'd like to thank my collaborators. This work was done while I was a PhD student at Brown, and it's being continued by one really talented undergrad, William Zhang. It was funded through a data science initiative seed grant, and the source code for the simulations and the analysis is available on my GitHub page. Thank you.
All right, so let's briefly unmute ourselves and applaud for the speaker. So we had a lot of good questions throughout, which I appreciate, but so long as Dhananjay is able to stay, we have time for more questions. So more questions from folks? I wanted to ask one question, Dhananjay. So your first model of motion, I'm a little bit familiar with the Dizornia model, right? And it has this large scale attraction, short scale repulsion. It's also in 3D typically instead of 2D. But what are the primary differences between that first model and the Dizornia model?
I think the key difference between the model I had and the Dizornia model is that the Dizornia model doesn't have the indicator variable. So cells, the long range attraction actually extends all the way to infinity, which means that if you start, no matter what initial condition you start with, you are very quickly going to formulate a swarm. There is one small set of parameter values in the Dizornia model where all particles go to infinity. So that's one where they do not form a cluster. But in the cases where they form a cluster and start swarming, that happens quite quickly because the attraction extends to infinity. The Dizornia model in 2D always gives a single phenotype. So it forms, a milling pattern or a double milling pattern. But if you actually simulate the Dizornia model in 3D with a little bit of noise injected into the differential equations, you turn it into a stochastic differential equation. That has more challenges because that 3D models will not directly go to whatever steady state is going to go into. It depending on how much noise you inject, it can actually switch stochastically between different phenotypes. So it's going to form a single milling pattern and then stochastically switch to a double milling pattern, then back to a single milling pattern. So there's the phase transitions happen over time, and that's quite tricky to incorporate. And that's kind of why we need to have we need to compute persistence not just at the final time step, but over time to be able to recover model parameters in that setting.
Any last questions?